Ultrafast protein folding pathways examine how proteins can fold into their three-dimensional shapes in an incredibly short time, even under extreme conditions. Proteins are essential molecules, and their function depends on their structure. When proteins fold correctly, they perform critical tasks in our cells. Under extreme conditions, like high temperature, pressure, or chemical stress, these folding processes can happen much faster than expected. How do proteins manage such rapid changes? The secret lies in the many pathways and intermediates they explore during folding. One example is when scientists observe protein folding using ultrafast laser techniques. These methods capture events that happen in just a few picoseconds, trillionths of a second. Researchers have found that proteins sometimes take unexpected shortcuts, forming stable structures much quicker than traditional models predicted. This raises the question: why do proteins fold so rapidly under stress? The answer may involve a combination of intrinsic molecular dynamics and the influence of the environment, which together help the protein avoid misfolding. In some experiments, proteins exposed to extreme temperatures initially misfold, but then rapidly correct their structure once conditions improve. This dynamic behavior is crucial for cell survival under stress. For instance, some organisms live in high-temperature environments, and their proteins must fold correctly despite these harsh conditions. These ultra-fast folding pathways ensure that proteins quickly reach their functional state, which is essential for maintaining life. Isn't it remarkable that nature can design proteins to work reliably, even in the most challenging environments? Another intriguing aspect is the use of computer simulations to map out these ultra-fast folding events. By modeling protein behavior at the atomic level, scientists can observe how slight changes in environmental factors speed up or slow down folding. These simulations help answer: what role do specific amino acid sequences play in the speed of folding? In many cases, certain sequences have evolved to form stable structures more quickly, serving as efficient nucleation points that drive the entire folding process. In summary, the study of ultrafast protein folding pathways reveals how proteins can rapidly achieve their functional shapes under extreme conditions. By combining experimental techniques with computational models, researchers have begun to uncover the shortcuts and rapid transitions that proteins use to fold correctly. Could these insights lead to new ways of designing synthetic proteins or treating diseases related to protein misfolding? The ongoing research in this field holds promise not only for our understanding of basic biology, but also for practical applications in medicine and biotechnology. Proteins are long chains of amino acids that must fold into specific three-dimensional structures to perform their functions. Protein folding is a rapid, dynamic process that can occur in microseconds or less under certain conditions, and ultra-fast folding pathways reveal how proteins achieve their functional shape quickly, even under extreme environments. Understanding these pathways helps scientists decipher how molecular changes affect biological activity. In laboratory experiments, proteins exposed to sudden temperature changes can rapidly fold into a stable structure within a fraction of a second. It's like watching a piece of origami being folded in a flash, where each fold happens so quickly that the final design appears almost instantaneously. Extreme conditions such as high temperature, pressure, or chemical environments can accelerate protein folding processes, with proteins potentially folding faster or following alternative pathways compared to normal physiological environments. Under these conditions, proteins may fold rapidly or via different routes. And studying folding under extreme conditions helps reveal the limits and adaptability of protein structures. This research is important for understanding how life can survive in harsh environments. In experiments, proteins in high-pressure chambers have been observed to rapidly assume their functional shapes, despite conditions that would normally destabilize them. It's like a metal spring that snaps into shape instantly when exposed to a sudden force, even if the force is much higher than usual. Ultrafast spectroscopy is a technique used to capture the rapid changes in protein structure as they fold, by employing extremely short laser pulses to record events that happen in millionths or billionths of a second. This method allows scientists to observe transient intermediate states that occur during folding, and the data gathered from these studies help build models of protein dynamics and function. Researchers use ultrafast laser pulses to capture snapshots of a protein as it transitions through various intermediate forms before settling into its final structure. It's like taking a high-speed video of a hummingbird's wings in flight, allowing you to see movements that are too fast for the naked eye. Protein folding pathways are often visualized as energy landscapes, where the protein moves downhill toward its most stable low-energy state. These landscapes include multiple pathways and intermediates, each representing a different folding route. Ultrafast folding studies help map these landscapes by showing how proteins rapidly overcome energy barriers, which is key to predicting how changes in conditions affect protein function. Scientists have created energy diagrams that show several possible folding routes, each with a different pathway from the unfolded to the folded state. It's like a mountain valley with multiple trails leading down to the base. The protein chooses the most efficient route to reach the lowest point. During the folding process, proteins often form short-lived intermediate states that are critical for achieving the final structure. These intermediates can provide clues about potential folding errors and how proteins avoid misfolding. And by studying these transient states under ultrafast conditions, researchers gain insights into the mechanisms that ensure proper protein function. In controlled experiments, scientists have observed brief intermediate conformations before a protein fully folds, which can help explain why certain misfolded proteins lead to health issues. It's like watching the various stages of a puzzle being assembled. Each intermediate step is essential to eventually see the complete picture. Molecular chaperones are specialized proteins that assist other proteins in folding correctly and quickly, especially under stressful conditions. They help prevent misfolding and aggregation by stabilizing intermediate states and guiding the folding process, playing an even more critical role under extreme conditions to ensure proteins reach their proper structure. In lab settings, adding chaperones to a solution of unfolded proteins can dramatically increase the speed and accuracy of the folding process. It's like having a skilled coach who helps an athlete perfect their technique, ensuring that every movement is executed correctly even under pressure. Understanding ultrafast protein folding pathways has significant implications for biotechnology and medicine, as insights into folding mechanisms can lead to new treatments for diseases caused by protein misfolding and contribute to the design of stable, synthetic proteins for industrial and therapeutic applications. Researchers can use knowledge of folding pathways to engineer proteins that are more resistant to misfolding, which is beneficial in designing drugs for neurodegenerative diseases. It's like learning the assembly instructions for a complex machine. Once you understand how each part fits together, you can build a more efficient and reliable version of the machine. Advances in technology, such as ultrafast lasers and sophisticated computational models, have revolutionized the study of protein folding by allowing scientists to observe rapid molecular changes and simulate the folding process with high accuracy. These tools enable detailed mapping of folding pathways and energy landscapes and are essential for translating basic research into real-world applications in medicine and materials science. Modern ultrafast spectroscopic techniques can capture every fleeting moment of a protein's transition from an unfolded to a folded state. It's like using a super high-speed camera to capture the intricate dance of performers on stage. The advanced technology reveals details that are otherwise invisible to the naked eye. The study of ultrafast protein folding is paving the way for deeper insights into molecular biology and the treatment of diseases linked to misfolded proteins. Future research aims to integrate experimental observations with computational models to predict folding behavior more accurately, potentially leading to breakthroughs in creating synthetic proteins and novel therapeutics. Understanding these rapid pathways could revolutionize drug design and our overall comprehension of molecular biology. Researchers are developing combined experimental and simulation methods to predict protein behavior under various conditions, which could revolutionize drug design. It's like assembling a jigsaw puzzle with the help of both a picture guide and a computer program that predicts where each piece goes. Together, they provide a complete and clear picture of the process.